Hi guys, just a quick video here on all those chromosome names. We keep identifying the chromosomes and calling them different names based on where they're at in the cell cycle and how they look to us. So I want to make sure that you understand that because it is important to know that information. All right, so here's our cell cycle. The blue is mitosis. This first step here is G1. And down here we've got G0 where we can exit the cell cycle. So we have gap 1, synthesis, and gap 2. Okay, so if we use this cell cycle to look at how chromosomes look during each step, I think it'll help you understand their names. All right. So let's start here in G1. Let me push this up and zoom in a bit. So in G1, the cell was just made from mitosis. So the chromosomes that are in the nucleus would be, if we were to look at it right then, I would say that it would look like chromatin, or it would be chromatin. Chromatin is loosely arranged. It's loose. It is being used at the moment. It's open. It's like a library that is available so that any of the DNA that needs to be read can be read and used to make RNA. So at this point, if we were to look at the cell, like that, this is my drawing of it. It would just be some loosely arranged. It would kind of be spread all around, different colors of blue if I can get some in here. Just kind of mottled looking, that bluish hue. Nothing really organized, right? It's organized as far as the cell needs it to be organized, but it's not condensed, it's loose, it is chromatin. If you were able to actually look at one molecule of DNA, you would find that there is only one copy of each chromosome at this point, okay? During this phase, you would find only one copy of each chromosome. One copy of each chromosome. Okay. All right, so the cell's going to go about doing his job and possibly going into G. This is messy. Let me make sure that looks like G sub G. G sub 0. Okay. He's going about doing his job, and he needs that chromosome to be loose so that it can be read. All right, so now in comes a signal that says, buddy, it's time to divide. You gotta go through cell division, so get ready. So what does it have to do to get ready? It has to go through synthesis, so it has to replicate its DNA. So during synthesis, DNA is replicated. And so the chromosomes go into a duplicated state. They're duplicated, and they are copied. Remember the semi-conservative replication. Each new DNA molecule is composed of half of the old DNA and half of new DNA. So we've got these duplicated chromosomes, and they like to hang out together. They will be organized. This cell knows that it's going to go through cell division, so it's beginning to organize itself. Just like if you are planning on moving out of your house, you organize your stuff and start packing it up. So that's what's happening here. This cell is organizing. So it takes those two identical DNA molecules that were just made and puts them together. Okay? So now these two identical chromosomes are together. They're exact copies of each other. We call these guys, or gals, we'll call these gals, sister chromatids. Okay? They are identical in every way. Identical copies. Okay? All right, and they are joined at their centromeres there. Very good. So at this point, we'd say we have sister chromatids. All right. Now, all along the way here so far, I didn't use this word and I really need to. All along the way here, I say you have only one copy of each chromosome, for example, here, one copy of each chromosome. Well, that's true. If this is a human, this cell would have 46 chromosomes, right? It has 46 chromosomes. Where did those chromosomes come from in that cell? 23 of them came from mom, and 23 of them came from dad. So chromosomes that carry the same information, let's actually do this in a different color. Do a different color here. We'll make pink for mom, and we'll do blue for dad. Okay, pink and blue. So we're going to say that these are identical chromosomes as far, excuse me, not identical. They are the same number chromosome. They're not identical because they're going to be slightly different in the genes that they carry. They're going to carry the information for the same genes, but maybe not identical information. So what does that mean? Well, let's say that this mom's chromosome, we'll say that this is chromosome number 2, okay? And on chromosome number 2, maybe you find a gene for a particular eye color. And I'm making this up, okay? And we'll say on mom's, it's blue, okay? And on dad's, the eye color gene says it's going to be brown. Well, they're both carrying, these are both carrying information about eye color, but it is slightly different, the information. So chromosomes that carry similar information, but one of them came from mom and one of them came from dad, we call these guys homologous chromosomes. Homologous chromosomes. Okay, so right now in this cell, in your body cells, each body cell that you have has 46 chromosomes, 23 that you got from your mom and 23 that you got from your dad, and you can pair them up. You can pair up homologous chromosomes so that you can see which ones go together. We call that a karyotype. We looked at in class, right? So here we've got our homologous chromosomes. Good. Over here we've got the sister chromatids. Now this is just one of these guys because we have to copy all of our homologous chromosomes. So if this was a, let's make this guy pink. Oh, I can do that. Let's color him. Okay, so let's make this one. This one's the pink one from mom. Okay. And don't forget, if we're going to do homologous pairs, we needed one from dad too. So here's the one from dad. Okay, and we're going to make dad's blue. Okay, there was the original chromosome. Okay, and we took half of him and made it new, and take the other half and make it new, and we just replicated our DNA. Okay, so now we've got sister chromatids here, but we also have a homologous pair, homologous chromosomes. So these guys are homologous. Because we got one from mom and one from dad. Okay. All right, as we go into G2, there's no change in what the chromosomes look like. Okay. And then when we go into mitosis, the chromosomes, the sister chromatids will be split. Okay, so that you end up with one cell, here's another cell. We go through mitosis and cytokinesis in my picture here. So now we're going to add chromosomes to these cells. So each cell is going to get one pink, and each cell, because we're splitting the sisters, each cell is going to get one blue. Just like we started with, one pink and one blue. Now that happens for all 46 chromosomes in a human cell, or whatever the chromosome number is for that critter. The sister chromatids are split during mitosis. Okay? We didn't change the chromosome number. The chromosome number was, 46 to begin with. We replicated our DNA and we split it, so in the end we ended up with 46 chromosomes in our offspring. Now I only drew 2, okay, here, but still you can imagine if there were 46, I'd have to have an awfully big picture. Okay, so that's it for all those chromosome names. Make sure again that you're familiar with sister chromatids, homologous chromosomes, the term chromatin, the fact that sister chromatids could also be referred to as duplicated chromosomes. Okay. Duplicated should be, maybe we should draw our arrow right there. Okay. My bracket is showing the homologous chromosomes there. All right, if you have any questions, let me know. Have a good one.